Thank you very much. Thank you, James, for that lovely introduction. And thank you, everyone, for being here. It's fantastic to look out and see friends and colleagues, former PhD students and current PhD students. But also, I'm blown away that we have a lot of the diabetes support groups here this evening. It's fantastic to have you here. Thank you so much for coming along. So I look forward to telling you a little bit about diabetes, to microgravity and beyond, but also the chance in my inaugural lecture to talk a little bit about my journey as a scientist. My journey began, I know, as a small ginger child in Glasgow, and I could not have imagined at that point standing here as a professor of diabetes and metabolism. So I'll tell you a little bit about that journey, but first of all, why diabetes? Well, diabetes is the most common metabolic disorder in the world. The World Health Organization tells us there are 500 million people with diabetes by 2030, and that's just the people that have been diagnosed. There'll be lots more that are undiagnosed. What that means in real life at the moment is about 1 in 10 people have diabetes. So if you have a look along your row, at least 1 or 2 of the people in your row, not this section, where the numbers are going to be a little bit higher, about 1 in 10 people now around the world has diabetes. So since I've started speaking, we spent somewhere between £250,000 and £500,000 treating the complications of diabetes. So there are large numbers of people, and the numbers are increasing. There are 2 forms of diabetes that my research career has focused on. Type 1 diabetes. Type 1 is an autoimmune disease. It's where the immune system turns on the insulin-producing cells of patients with that condition, and it leaves them dependent on insulin injections, usually from a very early age. The other form of diabetes that I've been focusing on is type 2 diabetes. Type 2 is the more common form of the disease. About 90% of people with diabetes have type 2. Type 2 is a hugely complex disease. It's influenced by our genetics, it's influenced by age, it's influenced by lifestyle. But both type 1 and type 2 are essentially not having enough insulin or not having insulin that's working as well as it should be. And the insulin molecule is at the center of everything I've been looking at throughout my research career. I was reminded of an old poster when I was putting my slides together, and I thought I'd show you that poster. This came out about 30 years ago, before most of the PhD students in the room were even born. Insulin is not a cure for diabetes. It just keeps people alive until we find one. It's something that's resonated my entire career. I'm going to ask you the same question I ask our undergraduate pharmacy students. If you don't have diabetes, I'll ask you to imagine what it's like to be diagnosed with, say, type 1 diabetes. And you have your insulin pen, and you have your blood glucose testing strips, and you have your carb counting book and your instructions on how to do all these things. And now you leave the clinic, and it's up to you to manage your own metabolism. If that doesn't sound scary enough, then I will ask you to imagine that it's not you that's been diagnosed with type 1 diabetes. Maybe it's your 5-year-old daughter that's just been diagnosed. And now it's your job to look after their metabolism, to learn how to do insulin injections, to do the carb counting, and all of these different things. It's challenging to manage your blood glucose if you have diabetes. What makes it more challenging is if you look around this room and look at the people on either side of you, your metabolism is completely different to theirs. So your metabolism is unique to you, for every single person in the room. How you respond to glucose, how you respond to foods, to drinks, how you respond to exercise, that's unique to you. And how you respond to your insulin is unique to you too. So this is a challenging disease. Everybody has a unique metabolism. And it's something we learned about a long time ago during our childhood. My influence at those days, this is my young mother at 18, doing her nursing training. And Mum, obviously, quite a scary looking syringe in this photo. This is Mum doing her training. Mum worked night shifts when we were children, and when she retired from nursing, she went on to work with deaf-blind children. And she brought us, myself and my 2 sisters who are here tonight, up to understand the uniqueness of individuals. Everyone's metabolism is unique to them, and to celebrate everyone's uniqueness. What she also taught us about was communication, and I was thinking tonight about linking things together throughout my career. And what I was thinking about in the end was communication. what I've been involved in, what I've been championing all these years, is really communication. Communication between people, but also communication between cells. So I'll introduce you to the cells I've spent my life looking at. These are the insulin-producing cells within your pancreas. So on this side here, we have a section through a human pancreas. These red cells you can see in the middle, these are the insulin-producing beta cells. On the left-hand side is my cartoon of an islet that I use in my teaching. Each of these insulin-producing beta cells, these are exquisitely sensitive to any change in your blood glucose concentration. As you're sitting here just now, if you've eaten a meal or you've been in the BrewDog having a quick pint before you come to the lecture, all of these things, your pancreas and your islets will respond to instantly to get your blood glucose back into that healthy range. So the beta cells respond to any change in your blood glucose to bring the glucose back down again, secreting insulin that targets all of the tissues in your body. Unfortunately, for people with diabetes, these are the cells that are targeted. So in type 1 diabetes, these cells are destroyed by the immune system, and it leaves people dependent on insulin injections. In people with type 2 diabetes, the beta cells are still there, they're still trying, but they're facing a lot of metabolic challenges. So these beta cells are fundamental to the whole thing. And how the cells talk to each other turns out to be really, really important. And that's what a lot of our work is focused on, especially since I moved down to Brighton, is how these cells communicate with each other. So communication is a theme running through. My communication skills, they had to start pretty early. So these are the early days of communication skills. You can see, look how cute Sister Sue is. Tiny Sister Sue. This is my older sister, Jill. You can see how proud they look that I've used this photo. Classic 70s hairstyle. And classic 70s wallpaper in the background. So we grew up in a one-bedroom tenement flat in the East End of Glasgow. So you learned communication quite quickly. Also an element of diplomacy and perhaps peacekeeping, I think. A little bit of peacekeeping. And for the times that the peacekeeping didn't work, picking the perfect sword. So this is me on holiday on Arran with Dad. We went every year to Arran, and I'm reliably informed I spent somewhere between 1 and 2 hours on the first day of the holidays picking the perfect sword, because you don't want to jump in with these decisions. And I think this is a good metaphor for life in science, and life in academia. So communication, diplomacy, peacekeeping, and if all that fails, pick the right sword. So that's something I learned at an early age. I became fascinated with metabolism. I loved sciences at school. When I finished at school, I stayed in my hometown of Glasgow and went to Glasgow University. So here's a lovely picture of Glasgow University, a picture also of my beloved Celtic. And here's Dad and I and Sarah heading off to Celtic Park to see the boys. At that time, I stayed not at home, but with this lady. This is Grace Macfarlane, my grandmother. Not because my diplomacy at home didn't work, but because my gran was a very influential figure. She was quite a ferocious woman, quite a fierce woman, but she taught us the power of learning, of studying, also how to cheat at cards. So she was a complex character, but she believed in hard work and she believed in learning. And she was one of the big supports for going to university. Not many people from my school had gone to university. It wasn't something we had a lot of experience with. So I lived with Grace Macfarlane for the first few years at university. And I learned about metabolism. Now, in those days, Glasgow was the heart attack capital of the world. And it was that for a number of reasons, high levels of smoking, but also the diet of the typical Glaswegian. When I went to Glasgow University, they started to teach us about metabolism and how all the different elements of your metabolism talk to each other. But we were also studying diet. So I will present you with this. No answers from the Glaswegians in the room. If you're not from Glasgow, can anybody take a stab at what this is? It's a deep fried. Too many of you know the answer to that. Far too many. Okay, so yeah, this is a deep-fried Mars bar. Glasgow has given the world art and architecture and literature, but we're famed for the deep-fried Mars bar. If you haven't seen a deep-fried Mars bar before, well done you. It is exactly what it says on the tin. It's a frozen Mars bar that's coated in batter. and then it's deep fried in beef dripping. The chip shop that we lived opposite when we grew up in Glasgow used to sell a deep fried Mars bar supper. And that was 2 deep fried Mars bars with chips. So it's not an enormous surprise that Glasgow is the heart attack capital of the world. It was at that point. But also a lot of diabetes. So I became obsessed at university by what all these dietary influences were doing to my beautiful little islets and the perfect beta cells with that exquisite sensitivity to any change in your blood glucose concentrations. So I completed my degree in molecular biology, trying to understand how everything was talking together. And at the end of that, I moved south of the border to the University of Birmingham to look in much more detail at what was happening to these islets of Langerhans. There's a nice picture of the Queen Elizabeth Hospital in Edgbaston in Birmingham, as it was when I was there. We knew at that point that the islets made insulin in response to glucose, and that insulin brought your blood glucose levels down. But we didn't really know much about what was happening inside these beta cells. So the question I was set as my PhD project was, how does glucose switch on the insulin gene? That was the subject for my research for the 3 years I was in Birmingham. Now I'll show you my little schematic of what goes on inside each individual beta cell. So here's what happens when you have something to eat and your blood glucose rises. Glucose goes in through a glucose transporter. It's metabolized. We know that gives energy to the cell in the form of ATP. ATP closes these potassium channels, depolarizes the membrane, and opens voltage-gated calcium channels. So it's the influx of calcium into the cell that triggers the release of insulin. All of this is happening as you're sitting here tonight. All the time, little puffs of insulin are being released to keep your blood glucose in that nice, healthy range. And all of these things must be intact and working for you to have normal glucose regulation. What we didn't know at that stage was how you replaced that insulin. There are some little secretory granules that sit right underneath the membrane, and as soon as you eat something, they release insulin. But those have got to be replaced so that you're ready to do that again the next time. So the question was, what was happening in the nucleus of the beta cells? How was the insulin gene being switched on when your blood glucose levels went up? So I'll fast forward 3 years and tell you the answer to the conundrum. And the answer is the obsession of my life, which is the protein called PDX1. If you only remember the name of one protein for conversational diabetes research, PDX1 is the one to drop into conversations. So PDX1 is a transcription factor protein, and it activates your insulin gene. Here you see some insulin-producing beta cells in the cell culture laboratory. The little green signals that you can see, that's the PDX1 protein stained green, and it's sitting around the outside of the nucleus. So in low glucose, it's sitting resting. It just sits on the outside of your nucleus, and it waits, ready for you to eat something. When you eat something or drink something and your blood glucose rises, it moves from the outside of the membrane into the nucleus, and it activates your insulin gene. And you can see here the red color, that's the production of new insulin that occurs when PDX1 moves from the cytoplasm to the nucleus. So that's 3 years of research in one slide. But PDX1 was the answer to the question. And here you see a freezing graduation day at the University of Birmingham, my frozen mum and dad down in Birmingham for graduation. So I became more and more obsessed with PDX1. What happened then is my PhD supervisor, the fantastic Kevin Doherty, got the chance to move the entire lab. So instead of being in the south, we moved even further north than I'd been before, up to the University of Aberdeen. This is me and Kevin. Back in the old days, you could settle things in a proper manner. So, this is Kevin, this is me. This is actually, was at the European Islet Network in Sweden, where we did a lot of work. We used to do the UK versus the rest of Europe. Who would know that was a premonition of things, to come. But he was a fantastic supervisor. We worked in Marshall College up in Aberdeen, which is a beautiful building. And Kevin took up the MacLeod Smith Chair of Biochemistry. For those in the diabetes field, MacLeod was, along with Banting and Best, the chap that won the Nobel Prize in 1922 for the discovery of insulin. So it was a very prestigious move for Kevin, and he moved us all up to Aberdeen. We got a huge program grant from the Wellcome Trust, and we built a complete obsession with PDX1. So I had 5 years postdocing in Aberdeen looking at PDX1. There was a fantastic team up in Aberdeen, all working on different aspects of islet biology. This is us in Connecticut, and this chap here became very important in my life and still is. That's Professor Jim Shaw. He becomes an important player later in the story. But Jim was there in Aberdeen when we moved up there. So my research as a postdoc was into PDX1. We knew that you needed PDX1 to move between the cytoplasm and the nucleus to produce insulin properly. So we started looking to see if maybe people with type 2 diabetes, maybe their PDX1 wasn't working properly. Maybe that was what the problem was. So we went over and worked in Sweden with a group out there, and we started to sequence the PDX1 gene in people with type 2 diabetes. And I'll show you one molecular biology slide. I couldn't help myself. A little molecular biology slide. Here on this axis you have insulin gene activation, so how much switching on of the insulin gene there is. And across the bottom, there are different mutations that we found in the PDX1 gene in people with type 2 diabetes. So the first column is how much insulin gene activation you get in low glucose, so not very much. In red, you see what happens when we add glucose, so you get a big activation, big production of insulin. And that's if you have the normal, healthy, what we call the wild-type PDX1. So if you have normal, healthy, wild-type PDX1, you get a big activation of your insulin gene. These are 3 mutant versions of PDX1 that we created that look like the proteins in people that we found that had these mutations. So if you have a cysteine to arginine mutation at position 18, here you see you only have about 70% of the activation of the insulin gene compared to the wild-type protein. Likewise, if you have a mutation at position 76, you get about 70% activation. And then quite a severe mutation at position 197 that changes it from an arginine to a histamine amino acid. And that gives you only about half as much insulin. And this was big for us. This was the first time that mutations in individual proteins, mutations in individual transcription factors, had been shown to be a causative factor in type 2 diabetes. So it was an exciting time. We did a lot of exciting work up in Aberdeen. And after my 5-year postdoc in Aberdeen, I got the chance to move south again, but also to start my own research lab. So I took my obsession with PDX1 and I moved back down south, but this time to Newcastle. You can see here the Millennium Bridge, which was built when we got there. So I went down there just at the Millennium, just at the start of 2000. 2 very big things happened in the year 2000, for me personally, but also for people in the field of diabetes research. So first of all, on a personal level, I was lucky enough to be awarded a Career Development Award from the Juvenile Diabetes Research Foundation. They're now called Breakthrough Type 1, a fantastic American charity supporting research into type 1 diabetes. And that was back in 2000, when $500,000 was a lot of money. So there was a lot of things that you could do with that. I was able to do 2 things. First of all, I started building my own lab. I had a fantastic postdoc that came down from Aberdeen with me, Susan Campbell. And we started to look in more detail as how the PDX1 gene was regulated, what was going wrong in patients with type 2 diabetes with this protein. But the money in those days also gave you the chance to step away from teaching, from the administrative side of the job, and just concentrate on doing your research for 5 years, which was fantastic. It meant I was able to build my own research group. They're so young, the young PhD students that started with me in Newcastle. And it also meant we were able to apply and receive funding from a lot of different funding bodies. So it allowed us to build up a research group and to start to talk about our research and to start to build collaborations and talk to other scientists and clinicians. My head of school in those days in Newcastle was a guy called Steve Yeaman, who was an ardent Newcastle United supporter. So when new staff started, we all went to St James's Park every Saturday afternoon to watch Newcastle United. We went to see them as a lab every week for 5 years. In that 5 years, they won nothing. Nothing at all. So I couldn't move on from this slide without showing you, yes, we finally won something. And this is the Newcastle United team very recently winning the English League Cup. So it didn't take long. It only took them 20 years after I left. to actually go in and win something, but I couldn't resist putting it in. So 2 big things happened. One, I got my Career Development Award. That was huge. It gave me the chance to build up my own research. The other thing that happened was much bigger in the world, because in the year 2000, the team in Edmonton in Canada produced the Edmonton Protocol for islet transplantation. It was always possible to have a pancreas transplant for patients with type 1, but it was usually only if you also needed a kidney transplant, and it was a very, very complicated operation. What the team in Edmonton did that was remarkable was they said, well, why are we transplanting a whole pancreas? All that's missing, all that's being destroyed in patients with type 1 diabetes are the insulin-producing cells. So they got donated pancreases, and they purified out from them just the islets of Langerhans, just these little 3-dimensional clusters of cells that are made up, the majority of them are the insulin-producing beta cells. And they reintroduced those into patients with type 1 diabetes using the simplest transplant operation we had seen. They put them in a syringe and they inject them into the hepatic portal vein. What happens then is pretty remarkable. They float through the hepatic portal vein, they lodge in the liver of the recipients, they take the blood supply from the liver of the recipients, and they start to function the way they do in your pancreas. They start to sense changes in glucose concentration. They start to secrete insulin in response to those changes. They essentially take over the normal metabolism and restore normal blood glucose control in these patients. And this was a game changer, an absolute game changer for how we thought about treating type 1 diabetes. It gave us proof of principle that if you could put the right number of cells back in there, you could essentially give people back the normal regulation of their blood glucose. So it was a huge step forward. And I say, it really changed how we think about treating diabetes. Here's one of the early patients undergoing their islet transplant operation. Here they are. In the syringe here are the donated, isolated human islets of Langerhans. They are injected through the cannula. What's strange is the patients are under local anaesthetic, so they're able to chat while they're having their transplant. Most of the transplants take 15 to 30 minutes, and most patients go home the following day. And no longer have the need for daily insulin injections, for blood glucose finger prick measurements, for wearing a blood glucose sensor, for carbohydrate counting, and all of the complexities of type 1 diabetes. So this was an absolute game changer. I'm really honoured that we have some of the islet transplant recipients here tonight. It means the world to me to have you here, so thank you very much for coming. This was a real game changer, and it led to the development of the UK Islet Transplant Consortium. Here's some islets, just to give you an idea of what size an islet is when it comes out the pancreas. Here's the needle tip that's used to deliver the islets. And all of this work led to the establishment in the UK of the first government-funded transplant programme for people with type 1 diabetes, the UK Islet Transplant Consortium. The driving force behind that was the man I mentioned earlier, Jim Shaw, an absolutely fantastic clinician and scientist. And I was very honoured to work with him in Newcastle and to still work with him. And he was the driving force behind that. So everything changed in 2000 because we were in the lab looking at PDX1. We were looking at cell signalling. We were looking at the minutiae of gene expression. And the drive around the world at that point really changed because now we had proof of principle. You could restore normal function just by putting in transplanted cells. There was a global effort to see whether we could generate new islets. We knew how to deliver them into patients. We knew that the patients would then have control of their own blood glucose, but we didn't have enough islets. There were less people on the donating islets list than we would need for even 1% of people to receive islets in a transplant. So we knew what the islets looked like. We knew how they functioned. We had worked on them for a number of years. We had looked at all the different things that stimulate PDX1. And PDX1 is a master regulator of the pancreas. Not only does it switch on and off your insulin gene, as it will be doing now as you're sitting here, but also it drives insulin-producing cell production. So we thought we're on a winner. We know what we're doing with PDX1. And we started looking at cells in the laboratory that we could turn into new islets. Could we create a multiple and abundant source of new islets? So we started looking at different types of stem cells. Here you see us looking down the microscope at some stem cells growing in the lab. Stem cell biology was also a very young field when we started doing this. And the team at Newcastle University were some of the pioneers of stem cell technology. Stem cells If you're not familiar with them, they're pretty extraordinary. We all start off as stem cells. And under the right set of circumstances, they can become any adult cell type in your body. The challenge, because beta cells are so specialized and so exquisitely sensitive to glucose, was to work out how to turn essentially a blank canvas of a stem cell into an authentic insulin-producing cell. So we thought, right, well, we know loads about PDX1. We know how it's switched on. We know how it moves about the cell. We'll take the stem cells, we'll put PDX1 in them, and they'll turn into insulin-producing cells. So I can summarize for you 5 years of research on this. Can we create new islets? No. No. What we were able to create were little clumps of cells. Some of them made insulin. They made it all the time. They didn't make it in response to glucose. So it was a frustrating 5 years. We made progress on understanding PDX1, but it's much more complicated than just switching on one gene. Your pancreas is a beautiful, exquisitely sensitive organ, and the islets are really fantastic at that. And it comes from lots of different cascades of gene expression. So we had to take a step back and learn what those cascades were. But we also had to think about how we were growing our cells. Cell culture, if you're not a molecular biologist, is done in most laboratories around the world by growing cells stuck to a petri dish, stuck in 2 dimensions, flat cells that grow in a dish. We harvest those cells, we divide the cells to more plates, and we have more and more flat cells. And that's how research was done for many years. But it's not how your cells work. Your cells work beautifully because they work in 3 dimensions, and they all talk to each other. And using the cell culture techniques we were using in our time in Newcastle, we couldn't reproduce that. So what we needed to do was think a bit more outside the box. And if you want to think outside the box, there's really only one place for you to come. So I moved my lab from Newcastle down here to the University of Brighton. Here you see my fantastic colleagues, Moira Harrison and Adrian Bowen, and we formed the Brighton Diabetes Research Group. As you can see, we thought long and hard about how to get the cells to talk to each other, how to get the cells to behave like they do in your body. We started looking at scaffolds. So here you see little green cells that are growing through scaffolds. We started to think about how the cells talk to each other. But the real breakthrough came, as is the title of the lecture, when we moved not from normal gravity, but into microgravity. So I'll introduce you to the research tool that's become such an important part of what we're doing. So we moved from normal gravity into microgravity. This is what a microgravity bioreactor looks like. This is what we grow and develop our cells in the cell culture room up at Huxley Building. If you haven't seen one of these before, each of these little individual pods, we put the cells inside the pod. They have a really clever polymer membrane in there that controls the oxygen tension, and it rotates at a really slow rotational speed. And what happens inside there is you create, through that movement, microgravity. So the cells are like the astronauts on the International Space Station. They go up, they float round, they come down. They don't touch the outside edge. They don't have any stresses on them at all. It's like a flotation tank for cells. The cells are relaxed and happy. And what happens when you make cells relaxed and happy is they behave like your cells. So they start to look for other cells, and they start to talk to each other, like your cells do in your body. And this was the big leap forward, getting cells to talk to each other. And I'll show you some of the original data, how we understood what the microgravity was doing. So we'll start in normal gravity. These are some electron microscope pictures. When you take a stem cell and try and turn it into an insulin-producing cell, the first thing you have to do is take them off of the plate that they're stuck to and turn them into the best 3D cluster or 3D clump of cells that you can. That starts the process of dividing. This is called an embroid body. So this is what happens if you try and make an embroid body in normal gravity. It looks a bit like a brain, I think, but it's tiny small. When we tell them to switch on PDX1, 2 or 3 of the cells will switch on PDX1, but not all of them. And the reason for that becomes clear, I hope. What we do is we cut this in half, and now we're looking in the middle. And here's what that looks like inside. So what I hope you can see is you can see each individual stem cell here. There's not a lot holding this thing together. And definitely what there isn't are cell-cell communication. the cells are not talking to each other. So we'll tell this cell to switch on PDX1 and become an insulin-producing cell, and it may well do that, but it won't talk to any of the other cells. So you end up with patches of insulin-producing cells instead of what you want, which is a whole cluster of cells that make insulin. So if you compare what happens in normal gravity with what happens in microgravity, hopefully you can see, first of all, when we generate the embryonic bodies, they're beautiful. They're beautifully spherical, and the cells are talking to each other. And I'll show you how I know that. So we're cutting this in half now, and we're looking on the inside. And here's what one of these looks like on the inside. So hopefully you can see, although you can still see little individual cells here, what you see in between these cells is beautiful deposition of something called extracellular matrix. All of this that you see in here is extracellular matrix. Now, extracellular matrix is what's holding you together. Without extracellular matrix, you'd be a big puddle of different cells on the floor. Extracellular matrix gives your organs structure. It tells them they're part of the same organ and they all need to work together. But what it also does is it allows the cells to talk to each other. It allows your cells to talk to each other and respond to things all together. So if we tell this one cell, switch on PDX1, become an insulin-producing cell, it will do that, and it will talk to every other cell it's in contact with in 3 dimensions and tell them to do the same thing. So just by switching on that one gene, we go from a clump of stem cells to an insulin-producing cluster. Insulin-producing cell clusters, these we call pseudo-islets. Looks like an islet, smells like an islet, produces insulin like an islet, but produced in the laboratory. These things are phenomenal. Here you see the beautiful Claire Marriott, who's here this evening. This is all Claire's work. This, I think, is a very moody photo of cell culture in the dark. I like it. So when you cut these pseudo-islets in half, and we look inside now at what's inside the pseudo-islet, hopefully you can see, you can't really even see individual cells. There's so much of this extracellular matrix. It's beautiful. If I took an islet from one of the pancreases in the room and took one little individual islet made up of the beta cells, if I add glucose to just one cell on the outside of that islet, we see that cascade of calcium go across the cells, and all of them release insulin at the same time. That's how amazing your pancreas is and how amazing your islets are. When we make pseudo-islets in microgravity, the same thing happens. We add glucose to just one cell and it goes glucose, and the signal goes across the whole islet and you start secreting insulin. So we made a huge leap forward by moving from 2 dimensions into 3 dimensions, and from moving from 3 dimensions into space, into microgravity, to be able to create the first clinically reflective model of a human islet. Something that behaved exactly like an islet. We had never had a model in the lab where we could look at these things, and we'd never been able to generate something that could function as a normal human islet for use in the transplant program. So it was a huge step forward. And a lot of the credit goes to Claire Marriott, who's here tonight. Other members of the team that were involved in working on the microgravity projects include the fantastic Sandeep Kumar. Sandeep's here as well tonight. Sandeep drove forward a lot of these microgravity projects with his imagination, with his passion for seeing what microgravity could do. The first challenge we had was, okay, you can make an islet, that's fantastic. You need millions. You need millions to be able to treat even one person, to give them enough insulin production capacity. So we started with little chambers, then we went up to medium-sized chambers, and then we went up to huge chambers. We thought at one point we're going to end up with things the size of a cartwheel, trying to grow enough of these things. But Sandeep's work was able to establish that these large chambers are enough to give us the insulin capacity that we need. So we're able to scale up this process. And I think when we started scaling up, we started to think, hang on, we have a model here that looks and behaves like an islet. So let's try and answer some of the questions that are outstanding from the islet transplant program. One of the challenges with islet transplants, when you first transplant them and they lodge in the liver, they've got to get a blood supply. So for a little while, they're in hypoxia. They have a low concentration of oxygen. And that was something we had never been able to study because we'd never been able to mimic that whole process. So what Sandeep and Claire and Noof did was they moved the microgravity chamber into a hypoxic chamber. This is a perspex chamber in our cell culture laboratory. The microgravity reactor goes inside it, and we can manipulate the levels of oxygen. The students call it the James Herriot chamber These are gloves under positive pressure. They blow out. So to be able to work them, you have to put your hand all the way into the shoulder, hence the James Herriot. You have to work with your hands inside it. What we're able to do then is to say, okay, well, your islets don't work at atmospheric oxygen. They're inside your body, so we take the oxygen concentration down. Then once we'd done that, we thought, well, the hypoxic islets, the islets that are being transplanted, that's even lower. And what this setup allows us to do is to mimic in the lab a clinically reflective model of what happens when the islets are delivered into patients. And it's the first time we've ever been able to ask questions about what's happening to the islets in hypoxia, what genes are regulated, how can we protect the islets from hypoxia. But Sandeep didn't stop there. He developed many, many more microgravity tools as part of the postdoc at the university. First of all, a chamber that allows us to mix cells together. So this is a perfusion microgravity bioreactor. You can see the little tubes going in one side and going out the other side. What it allows us to do is keep our cells alive in the microgravity chamber, but mix them with, say, cells from the immune system, or cells that help us grow blood vessels, and to keep them alive, not for days, as we can do in normal cell culture, but for weeks and for months. If we want to see what's happening to your islets, if you're someone with type 2 diabetes, and your blood glucose levels maybe vary a little bit, and we want to see what that does to the islets, this system allows us to keep our islets alive inside there and mimic all those fluctuations in glucose and see what happens. So it's an extraordinary tool. I think on a practical level, my very favourite bit of what Sandeep was able to create using microgravity was to answer a really practical challenge. The islet transplant program is the donation of a pancreas. The pancreas is then digested at an islet isolation centre, and the islets are transported to wherever the person is that's going to have the transplant. Traditionally, that's done the same way if it was a kidney or anything else, on the back of a motorbike in a chamber, on ice, kept cold. But the islets are very sensitive, and sometimes that transportation process is quite hard on them. So the challenge that Sandeep was given was to create a portable microgravity bioreactor, something we could use to take islets from A to B. And this is what it came up with. It looks like a picnic basket, but it plugs into your car charger, or you can keep it in an ambulance, obviously, on the back of on the back of a motorbike. It looks like a picnic basket, but inside it has a fully working portable microgravity bioreactor. So for the first time, we're able to generate the islets. We're able to put them in the microgravity where they float freely. They're completely stress-free. They're completely sterile. They're kept at 37 degrees, and they can be transported safely to wherever the tissue is required. So all of these practical steps forward came about from thinking in 3 dimensions and thinking about what's the next challenge. In diabetes, it's always the next challenge. You get to one point, and then there's another challenge. The very last challenge that we're working on now is how to protect the islets when they get transplanted. At the moment, if you have an islet transplant, you still need a certain amount of immunosuppressants to protect you from having the transplanted material. The holy grail for everyone in type 1 diabetes is to not have to take the immunosuppressants, to be able to transplant the cells and have them live a long and happy life in their new recipient. So the latest projects that we're involved with are how to protect the islets. We know how to generate them. We know how to transport them. We want to be able to transplant them without the need for the immunosuppressants. And for this, we started working with another team, this time Professor Matteo Santon, Gary Phillips, who are here this evening, and a fantastic postdoc, Valeria. And we started looking at a brand new field for me. I think that's the beauty of Brighton, is sometimes just chatting to people, you see how their technology is something that could help solve one of your problems. So I started working with Matteo and Gary and their group on nanotechnology, something completely different. So let me tell you what you're looking at here. Here, this is an electron micrograph of a slither of ice. Growing on that slither of ice are tiny nanoparticles of a substance called chitosan. Here we see an extreme close-up of the chitosan. These are little chitosan nanospheres. Now, chitosan is something that's used to coat medical implants. It's something that your body doesn't really react to. But what Matteo and Gary and their teams were able to do was to take this down in scale to the nanoscale. And in combination with the cells that we generated using our microgravity bioreactor, we were able for the very first time to coat the islets, not put them in a capsule, not put them in an implant, not put them in a chamber. Coat the living cells with what is an immunorepellent corona, a single nanolayer of material that repels the immune system, to hide these cells from the immune system. And this was some of the first work we did on that. I have 3 current PhD students who are here, who are now working on how to protect the islets, carrying on this work. How do we protect them? How do we allow them to function better and longer in patients when we've had the transplant? So microgravity changed the way we think about diabetes research, but for my title tonight, I put to microgravity and beyond. And I thought I'll finish with a little bit of beyond, because when you work on a clinically reflective model, when you start to talk to people about their research, and you start to talk about what you've got, you find yourself getting involved in a lot of different projects. And I thought I'd finish very, very briefly by telling you about some of the different projects that I've been involved in with microgravity and beyond. So not just diabetes research. This is Sandeep again, looking very fetching in his kilt here. This is work that Sandeep did with the chamber that I mentioned earlier that allows us to use microgravity to use that living cell model to see how cells interact. This time not diabetes though. Sandeep was working with a team up at Barts, and they were looking at pancreatic cancer. Pancreatic cancer is an extraordinarily aggressive cancer. And very little is known about why you get that aggressive growth of the pancreatic cancer cells. And that's because we've never really had models to look at that process. So what Sandeep was able to do, and you see some of the photographs of the interactions here between pancreatic cancer cells and our 3-dimensional islet models. It's the first time we'd really been able to look at the interaction between these cell types. So the model works just for diabetes, not just for diabetes, but for different diseases as well. Pancreatic cancer turned into something even more exciting. So whilst Sandeep and Claire and Noof were looking at hypoxia in the James Herriot chamber and looking at other genes that were regulated by hypoxia, so in the lab, Cressida Boya, who's here this evening, did a fantastic PhD looking at hypoxia, but not looking at the pancreas. So looking at hypoxia as a new drug target in liver cancer, another very aggressive cancer. So when you have these good technologies and when you talk to people, these technologies can be applied to a lot of different research questions. And I thought I'd finish with 2 of the more beyond from diabetes research. One of the projects that we were able to contribute to, and this is Dr. Brian Jones, who's now at Bath, but he's here tonight, and a fantastic PhD student, John Zakibineo. And they were looking at E. coli, so they were looking at bacteria. But how cells talk to each other is how cells talk to each other. And good technologies will translate across fields. So you're looking here at how bacteria talk to gut cells. And what we were able to do using our cell models is to begin to understand how these cells talk to each other. Now, what has that got to do with diabetes? I hear you cry. Well, it's got loads to do with diabetes. There may be some people in the room, and certainly you'll have seen it in the news about Ozempic, Mounjaro, Wegovy. All of the new drugs for type 2 diabetes are based on GLP-1, a peptide that's produced by your gut cells. So fundamentally, in people with or without diabetes, how your gut cells talk to bacteria and how they talk to each other is fundamentally important. And cell models are allowing us to understand these things. I love the opportunities you get at the University of Brighton to work on different aspects of different projects. And I'm going to finish off with the most recent work that we've been doing with Bhavik Patel and his lab. This work's been done by Chloe Miller, Bhavik's PhD student, a fantastic PhD student. I know she's here somewhere. And Anisha, my PhD student. And they're looking at, I'm not even going to try and read it out to you. Prussian blue, anyway. They're looking at electrodes, and they're looking at 3D printers. And let me tell you why this is important and why I'm excited about it. What Chloe is doing, which is incredible, is she's 3D printing electrodes to form sensors. And what the recent paper showed was the square grid sensor that's the most important thing. Again, why am I telling you this? What has it got to do with diabetes? Well, here's the exciting bit. Because in the paper, we looked at how much glucose was being used and how you could measure it using a 3D printed sensor. So on the left-hand side in blue is where there's no cells, and on the right-hand side in red, This is when we've added HepG2 cells, which are liver cells. And we can measure the amount of glucose that's being used by these 3D printed sensors. And if you're wondering why that's exciting, then speak to the people in the room that have diabetes. Speak to them about finger prick blood glucose testing, about wearing glucose sensors all the time. Speak to them about every 6 months having HbA1c measurements done. If you're newly diagnosed, having measurements of C-peptide, having antibody tests, and all of these things that require hospital visits and clinical visits. What the development of this technology creates is the potential for 3D printed sensors that will make life a lot easier for people with diabetes. It also feeds my mania that all research is really about diabetes. You just have to look hard to find it sometimes. So it's really, really exciting research. So once you understand how cells talk to each other, you can apply that to lots of different fields. And I guess that's my conclusion tonight is cells will always find each other and they'll always talk to each other. So here you're looking at a lovely EM photo. These are 2 beta cells. The first one started down here. But they send out signals. And slowly but surely, the other cell will find it. Cells will find each other, and cells will talk to each other. And I think that's true also of people. Good people will find each other. Good scientists will find each other. But good people certainly will find each other, and you'll find the right person. The biggest thing that's happened to me in Brighton, well, I found my right person. So this is my beautiful wife, Sarah, who's looking mortified. Fantastic. And this is our wedding up in Glasgow about 3 years ago now. This was our 3rd attempt at the wedding. The first 2 were very selfishly derailed by a global pandemic. We actually got married the week they lifted the COVID restrictions in Scotland. So you see, she is Vanessa. She's still got her Macfarlane clan face mask on. And I think if you look hard enough, you can see Shawnee. He's got his Brighton and Hove Albion face mask on. So it was a fantastic day up in Glasgow. And I do believe that Brighton's the great place and people will always find each other. So I'm going to finish off with my thank yous and the people that we couldn't do the research without. Hopefully I've name-checked a few of the stars as I've gone through. But research in Brighton, for me, has been hugely influenced by being able to interact with amazing patient groups. Some of them are here tonight, which is absolutely fantastic. And I wanted to thank them for their contribution to our research. We have the Brighton Area Group of Diabetes UK. They have been fantastic supporters of ours. Since the day I moved down here, they're absolutely fantastic. The Lewis Type 2 Motivational Group, absolutely brilliant. Support for diabetes in Burgess Hill. The young Type 1s, Elaine Sharp, who's here somewhere, couldn't have done her PhD without the extraordinary youngsters with Type 1 who took part in her PhD project. And the Hailing Island Diabetes Support Group, there they are, when we took down all our 3D pictures of what an islet looks like. Everybody looks cool when they put the 3D specs on. So we couldn't do our research without the input of patients and the families of people with diabetes, and it means an extraordinary amount, and it's fantastic to see so many people from these groups here tonight. I hugely appreciate it. I was chatting at the Lewis Type 2 Motivational Group a couple of weeks ago, and I know Jenny and Graham are here tonight. We were talking about motivation, of what motivates you, of how you stay motivated when you're having a bad day. No matter how good you are at controlling your blood glucose if you have diabetes, sometimes you have bad days. And you need people to support you, to talk to, and to help you through those. And I was thinking about the people that helped me. And these groups are some of the people that helped me. And I wanted to finish by thanking some other people that I really couldn't do any of this without. Many of you will know that before the pandemic, I'd spent my evenings and weekends working at Virgin Active as a spin instructor, indoor cycling. So this is me on the spin bike wearing my world record medal. Oh, yes. World record for our spin team. It was the most people on indoor cycles cycling at the same time around the world. So there was people in South Africa and all the way across Europe, and we all did it at the same time. We're still in the Guinness Book of World Records. That's just me, though. And in fact, there was a whole team of people. And I think that was my concluding thoughts on how to wrap up this evening, was to say, I do nothing on my own. Every bit of good science that I've done, wherever I've been, has been part of a team. Well, I've had some fantastic PhD students, academic supervisors, academic colleagues, and collaborators. But in my personal life and working at the gym and working with patients with type 2, these people motivate me. So I'm going to give a shout out to my team. Some of these are the fellow world record holders. They're all here looking mortified, yes, to see their photo up on the screen. Some of these are in the world record team. The spin class was not a traditional spin class. It was more dancing on the bike and messing about. And there's no better demonstration of that than Christmas spin. So this is Christmas spin. This is us all on the stage at the end of Christmas spin. Christmas spin started as just playing a couple of Christmas songs and putting tinsel up. But it turned into that you could see the spin studio from space. It had lights and trees and bells and whistles. And yes, you're not wrong, it is someone in an inflatable turkey suit. And if you've never seen someone doing spinning in an inflatable turkey suit, it's quite something to behold. This, is, as most of you know, is my best pal, Rocky. And Rocky and I have had a lot of adventures that have kept me going on my research side of things and in all other aspects. This is me on my 50th birthday with Rocky doing indoor skydiving. If you've never done it, I recommend it. It's like having a facelift. There's a lot of G. Rocky is a nurse, and she works with people with diabetes. And she's a great motivation and a great supporter of mine and always has been. Also a great person to rant to. We all need people in life that we can rant to. So a huge, huge heartfelt thank to my team and the people that supported me. Also, thanks to my academic colleagues. I'm going to finish off now. So many people to thank, everybody that's here tonight, basically. A thank you to all of my PhD students. These are some incredible scientists. I'm so proud of what they've all gone on to do. And so proud that so many of them are still in diabetes research. Absolutely fantastic. So a huge thank you to all of them. A big thank you also to all of my collaborators, principally among them James Shaw, of course, who is a phenomenon for the islet transplant programme and for his passion for diabetes. But all of these people, some are former University of Brighton folk that have moved on or have retired, but they've all been hugely important in the work that I've been doing. My current colleagues in the School of Applied Sciences down at the bottom, I can't put them all on a slide. I'd need about 15 slides, so you know who you are, and thank you so much for your help and support. These are my current PhD students, again, looking suitably mortified to see their photos up there. Maria, Anisha, and Monique, they're talking about their science, always talking about the science, fundamentally important. And I'm going to start as I finish as I started, rather, just by saying a huge thank you to all the support groups that are run by people with diabetes or by the families of people with diabetes. Your input means an enormous amount. It has since the day I moved to Brighton, and it will continue to be that way. And that is my journey, diabetes to microgravity and beyond. Thank you for listening.